So to review that, an unsaturated fat is a fat where not every carbon has all of the hydrogens attached to it, and that's because there are some double bonds. So there's some hydrogens that are missing, so hence the terminology unsaturated. It's not fully saturated with hydrogen atoms. Compared or contrasting that to a saturated fat where the carbons here in black are chemically bound to the maximum number of hydrogens, so fully saturated with hydrogens, if you want to think of it that way. And at room temperature, the unsaturated fats tend to be liquid because the double bonds form bends or kinks in the fatty acid tails, causing these triglycerides not to pack up so tightly together, causing it to remain a liquid at room temperature, compared to a saturated fat. Since the fatty acid tails are linear, they can pack up and line up more compactly the triglycerides in saturated fats, and because of that, they tend to be solid at room temperature. And as you may know, saturated fats tend to be less healthy for us versus unsaturated fats. Okay, so continuing, so most animal fats that we consume have a high proportion of the saturated fatty acids, and that causes them to tend to be solid at room temperature. And again, those are the ones that tend to be less healthy for us. They contribute to atherosclerosis. This is where you get plaque buildup in your blood vessels, and that can lead to further complications in terms of long-term health issues. So atherosclerosis is not a good thing. So however, on the positive side, most plant and fish fats tend to be high in unsaturated fats, and because of that, they are often liquid or very soft at room temperature, and then they tend to be more healthy, or the healthier fats. So let's move on to steroids. So steroids are unique. I was saying steroids are unique lipids because they have a unique structure that's different from other fats. And that structure, of course, leads to unique functions. So the carbon skeleton framework, instead of a linear structure, they form 4 carbon rings. So this is a shortcut diagram, but at every bend or kink, there's a carbon atom. And this is not showing you the hydrogens that would be bound to all of those carbons. So this is a basic steroid skeleton, 4 carbon ring structure. And depending what these functional groups are, changes the chemical characteristic of the basic steroid. For example, this is cholesterol. So the steroids vary in their functional groups, and that affects their chemical reactions and their functions and behaviors in the body. So cholesterol, for example, is a key component of cell membranes, and it's the base steroid from which your body will produce other type of steroids like estrogen or testosterone. So estrogen, which you can consider the female hormone, versus testosterone, the male hormone. One of the main hormones that produce feminine characteristics in animals, and testosterone, one of the main hormones that produces male characteristics in animals. So the base molecule is cholesterol, and notice the difference between testosterone and estrogen. There's an oxygen here and an OH there, and then an OH here and an oxygen there, and that's it. Those 2 functional group differences cause pretty significant differences in terms of the effects of these hormones on bodily tissues and organs, and of course on the entire body itself. So continuing with steroids, you may have heard of synthetic or anabolic steroids. These are steroids that are produced in a laboratory, and they're variations of testosterone, and they have been used and abused by athletes, even though initially they were generated to treat certain cancers and aid characteristics or aid symptoms in AIDS patients to have them live longer and to combat some of the debilitating effects of AIDS. However, they have been abused by some athletes, actually probably many athletes. They help build muscle quickly, but they can cause serious physical and physiological and mental problems. Okay? So basically, cheating by using synthetic anabolic steroids. Oh yeah, so if you've heard of roid rage in the sense of steroid rage, because of the effects of synthetic anabolic steroids on individuals, that has been documented to a certain degree. So let's move on to proteins. So proteins fall into the structural characteristics that carbohydrates have, in the sense that monomers are linked together to form polymers. With the proteins, the monomers are amino acids, and they get linked together into the polymer, or polymers that we refer to just as proteins. So proteins are very important. They account for over 50% of the dry weight of cells, and they function in almost everything cells do. So much so that when we talk about later on the expression of genetic material or genes, we're almost always refer to the gene product as being a protein of some sort. So major types of proteins. Proteins provide structural support. So your hair is made up mostly of protein. The horns on animals and your fingernails and toenails are proteins. What gives our skin the ability to resist abrasion and such are proteins. Our muscles are made up mostly of protein that do provide some structural support. So proteins are very important in that sense. Storage proteins provide amino acids for growth. So we have storage proteins that are broken down to form the monomer amino acids that then organisms can use to build the proteins that they need in the correct amount at the correct time. Contractile proteins in muscles help us move our bodies, move food through our bodies, movement. There's transport proteins. Transport proteins help transport substances within cells or substances through the body, like hemoglobin that helps transport oxygen. And then there's enzymes that help chemical reactions occur faster and faster and faster, speeding up the naturally occurring chemical reaction rate. And if you remember, I showed you a little video that showed how monomers can be linked together to form larger, like disaccharides or polysaccharides. And I showed you a little video where there was an enzyme that latched onto two monomers and then helped them link together. So those type of molecules that assist in forming chemical bonds are enzymes. And then the opposite is true also. Molecules that help break chemical bonds are other types of enzymes. So the monomers, they're amino acids. We have a total, or there are a total of 20 amino acids that are used to make proteins in humans. So amino acids, the monomer of proteins, they have a central carbon that's covalently bound to 4 groups. There's what's called the carboxyl group, the C and the oxygen here, carbon, oxygen, and an OH group. And then there's also an amino group, which is a nitrogen and 2 hydrogens. And then there's a lone hydrogen all by itself. So remember, carbon can form up to 4 bonds, 1, 2, 3, and then the 4th bond is a variable side chain, which is the functional group that's variable in the amino acids. So all 20 amino acids have the same amino group, lone hydrogen or carboxyl group, and what's different is this side chain, and that makes the different amino acids have different characteristics, chemical characteristics. Okay? So AA here means amino acid. Okay, so the overall structure and function, how protein structure is critically associated with this function. Right? So cells will build polypeptides, which become functional proteins, by linking amino acids together through a long series of dehydration reactions, forming peptide bonds. Peptide refers to protein. And as each amino acid is chemically linked together via dehydration reaction, there's the water getting kicked out, forming a covalent bond between the two amino acids. It's referred to as a peptide bond because, again, peptide refers to protein. And your cells do this hundreds and hundreds of times, up to thousands of times, to form a long chain of amino acids covalently linked together, and that's referred to as a polypeptide. Then the polypeptide is modified later within the cell to make the functional protein that will do whatever it's supposed to do in the cell. And some proteins get exported out of the cell. So a functional protein has at least one polypeptide chain that is eventually to form a unique specific structure, usually a globular structure, and that structure will allow the protein to do its specific function. Some proteins are made up of multiple polypeptide chains. For example, hemoglobin in your red blood cells that transports oxygen is made up of 4 separate polypeptide chains. It forms a nice big globular functional protein, the hemoglobin protein. So let's look at protein structure.
Most proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure.
And that's it. We'll talk about the 4 levels of structure as we continue. So how are the huge variety of the proteins made from just 20 amino acids? So only 20 amino acids, but a typical cell can make hundreds of different proteins. How? It depends on the sequence, the unique sequence of an amino acids in a particular protein. Like the English alphabet, if you think about that, there are 26 letters in the English alphabet, but depending on how the letters are linked together, will influence the resulting structure. Okay, so the word different, it's because it's D, then I, and then F, etc., and that makes the word different. Well, the 20 different amino acids, depending on how they're linked together in the appropriate sequence for the particular protein, will determine 1, the structure, and then 2, the function of that particular protein. Okay, so the proteins are made up of different proteins, of different polypeptides, because of the different sequence of the 20 different potential amino acids that are linked together through peptide bonds, again, to make the unique polypeptide that will become the unique and specific protein. Okay, so primary structure, right? Primary and first level of structure.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function.
Okay, so primary protein structure, the unique sequence of amino acids.